Welcome
Hi,
back
and welcome to
right
the AP
there.
Biology
I
Lab
don't
7
know. Is it a
Genetics and
correct? My
Drosophila
friends has me
podcast.
here.
This is Mr.
That
Anderson.
alone
And
here, alone.
in the
Look
past,
at
we
the
tried doing
bee.
the
There's so
fly lab,
many.
but
How can
great
it
success.
be?
And so
How can
I
it
went ahead and
choose?
breed them in
No,
class.
hang on, every
Usually
kid.
what
You
ends up
like
happening
your
is we have a
biology?
bunch of escaped fruit
Biology
flies
lab.
in the lab.
We
And
find
so
that
we used
clue.
the
I
virtual
don't
fruit
know.
fly
Is
lab.
it
And so where did the fruit fly
great?
lab all begin? It's with
Around
this
like
guy, Thomas
this
Hunt
ever.
Morgan. And so
Seems
he's in
like
his
this
fly
here.
lab right here,
I look
working on
like
this fruit
this.
fly. Fruit fly is great because it has
I
pretty
keep
simple
excited.
genome,
I want has
reproduces
me
very
so
quickly, and
bad.
it's easy to keep in the lab. And
Schoolgirl
so,
is huge. This
so
is
in this
our
lab,
right
let me
here.
kind of walk you through the way we
Science has
use
me here. We can't actually see it.
sciencecourseware.org. They've put
This is
together this
our
virtual
one
fly
here.
lab,
Male
and you can
and
really crank through a
female.
bunch of
Male and
fly crosses
female.
really quickly, and the statistics
There's
are
a
pretty
male
good.
and
And so
female.
first of all, we should talk a little bit
We
about
find
what makes a
him. I
female
don't
fly
know
different from a male
everyone
fly,
here.
because that's something on the AP
Watching
test
me
you
here
need to
alone
know.
here.
Female flies
Now,
in general
hang
are going to
on,
be a
every
little bit
kid.
bigger than
We're
male
getting away
flies.
from.
Male flies will also have, you can kind of see their rear
There's so
end is going to
many.
be a little bit
How
more
can it
rounded
choose?
than
I
the
don't know.
females, which is a little
Now,
bit
hang
pointed.
on,
And then
every
the big
kid.
thing that I always see is
There's
that the
a
males will have these
working
comb-like
one.
structures called the sex combs,
I
and they'll
want
sit on the
me.
front
That's enough.
feet.
I
And so
can
that's
go.
one way you can tell the difference.
Have
What
me
you do is
here.
you just
Don't
knock them
tell
out.
me
So you have this,
that.
you anesthetize them, and then they'll knock out. You can count them under a microscope, but we're going to do that all virtually. So let me jump into a browser for a second. And so you can log in as a guest, or you can set up a whole class. But let's log in as just a guest. And so in order to do a cross, we'll just do a simple one. You click on the computer, and then we order flies. And so it asks me, do I want to order a female fly or a male fly? So let's just order a female fly of the wild type. And so I just click on this add to cart. Whenever I'm doing the lab on here, I always get lost and look at the big yellow box, and it tells me what I should do next. And so if you ever get lost, click on that or look at that. So if I click on add to cart, now it's added one female, and now let's grab a male of a different, for example, let's try wing size. And so if we click on that, we've got choices, and so let's grab a male that has vestigial wings. And so now I'll buy him, and now I've got my cart filled up. So I just say done. Do you want to buy these? And I say yeah. So I'm going to check out. Alright, so now they arrive in the mail like this. I can click on the box, and it's going to put those in a mating chamber, and it's going to put them in the incubator. So now we just simply let them go. So 2 weeks later, then we get new flies.
Trouble. We'll find there's over 1960s. There's a primal swing blueprints at
the sound effects are kind of annoying.
We find
a
world
off, and the animations as well. There's
a right there.
Through a lot of classes. So let me grab this. So now we've got those crossed. So it was a female of the wild type and a male of vestigial wings. We now put them underneath that microscope, and then we sort the flies. So they would be asleep at this point, and then we're going to sort them out according to gender. And so it also could be any other phenotype. But if we look at our two piles here, we see we have 543 offspring that are female wild type, and then over here we have males of wild type as well. So it looks like we have an equal amount of each. Now that would be the F1. So let's do an F2 cross then. So I'm going to look at one of these flies. You can see this is a female. And I'm going to put her in a new mating. So she's added to a new mating jar. Let me zoom out for a second. Now let me grab one of the males. And I'm going to add him in a new mating.
There's your dirty.
And then finally I'm going to zoom out again.
Over here.
And now I go back to the lab. And you can see that it's added those to a new beaker. I throw them in the incubator.
Well, watch this. Probably one here more. There's one me. You're me here. The science.
Let me grab them out, and we'll look at those underneath the microscope.
One here. The science has me here alone. Actually, you know, has me. World largest for MFS has me here.
Alright, so let's sort those flies.
It has alarm.
And now we have different groups. In other words, we've got, it looks like 466 male of the wild type, whereas we only have 133 females of vestigial, 168 males vestigial, and then 443 females of the wild type. So this is kind of looking like that 3-to-1 ratio, that Mendel cross, that famous cross. We could actually send the data at this point to the computer, and you can do chi-squared analysis. I won't talk about chi-squared analysis on here, but I'll probably do that in a different podcast. fast. But what do you do at this point? You want to figure out what's the inheritance pattern. So it's looking at this point like wild is dominant, or a better way to say that is vestigial is recessive. So we kind of have that. We're going to look at, is it a recessive or is it a dominant trait? But then we also have on this matrix two other things. Is it an autosomal? Is it autosomal gene, or is it going to be sex-linked? And so the next thing I would want to do is I would want to do a reciprocal cross. So then I would do a male of the wild type and then a female of that vestigial cross, and then do point squares and try to figure it out. So that's the Fly Lab. Again, you can turn off the animations, the sounds, and you can really crank through a bunch of these processes really quickly. I hope that's helpful. Hi, and welcome to the AP Biology Lab 8 Population Genetics and Evolution podcast. In this podcast, we do what's called the Hardy-Weinberg Lab. Hardy-Weinberg, remember, is a way to describe in a population how the genes will change over time, or how the better way to say that is how they won't change. And so the reason I put a picture of Mr. Darwin here is that we can use Hardy-Weinberg equilibrium to observe when evolution is actually taking place in a population. And so what we're dealing with is microevolution. In other words, changes within the gene frequencies in a population. This graph is kind of confusing unless you really don't know what P and Q values are. And so P and Q values are going to be the frequency of the dominant and frequency of the recessive allele. And so as the number of big A, or homozygous dominant individuals, we'll say right here, increase up to a total of maybe 100% of them, you can see that the frequency of the other two phenotypes decrease. So if you're really into that, take a second to look at that. If not, look to the next page. So this is Hardy-Weinberg equilibrium, or Hardy-Weinberg equation is a better way to say that. And so first of all, we should kind of detail ourselves with what's down here in the middle. P-value is going to be the allele frequency of the dominant, and Q is going to be the allele frequency of the recessive. And so in this lab, we'll use a cup. We call it the mating chamber. And inside there, we're going to have beads. And those beads will either be black, and we'll say that's dominant, or they're going to be white, and we'll call that recessive. And so if I put 50 beads of black and 50 beads of white in the cup, what's my p-value? P-value is going to be 0.5. What's my Q value? Q-value is also going to be 0.5. And so in this lab, every time we start, we always start with a P and a Q value equal to 0.5, or 50-50, half of each. So what does the equation even tell us then? If we look at the equation itself, P squared, if we take P squared, that's 0.5 times 0.5, which is 0.25. What that tells us is the frequency of homozygous dominant. In other words, if I shake this up, the odds of me pulling out two beads that are both black should be 0.25. In other words, there's 25% chance. What about pulling out two whites? Well, that's also 0.25, or that's Q squared. And then what about the odds of me pulling out one that's heterozygous, either black-white or white-black? Well, that's going to be at 50%. And so if I shake it up and pull two out, what are the odds that it's going to be black-white? Should be half of the time. Now, I can't look, and so if I pull those two out, I got black-black. What are the odds of that? Well, it'd be 25%. Let me try it again. Pull it out. Black, so that's pretty rare. What about the next time? It's black-white. And so that should happen 50% of the time. Now that's an incredibly small sample size. And so in this lab, we have to make sure we pull a whole heck of a lot more than just 3 pairs. In fact, we pull 40 pairs out each time. And so what is equilibrium? Well, Hardy-Weinberg equilibrium is more of a mathematical model, and it was discovered by two different mathematicians at about the same time, Hardy and Weinberg. And this happens a lot in science. But what they said is if we keep these 5 things the same in a population or in beads in a cup, in other words, if there's no mutation, the beads don't magically change to a different color. If there's no selection, I'm not drawing out ones of a specific color to get rid of. There's no gene flow. I'm not like adding new genes to the cup, and I'm not pulling genes out. If it's a large enough population size, and if we do random mating, in other words, I shake it up each time and I randomly pull it out, it should stay at 0.5 that whole time all the way across. And does it ever do that? No, not really. But look at this graph, which is pretty cool. Over here, what we have is a population where the N value is 20. the N value is 200, and the N value is 2,000. N is the number of individuals in that population. And you can see that the bigger number we get, the closer it stays to that equilibrium. And as we decrease the number, then it's just going to get random. In other words, the law of large numbers says stats don't really work unless you have enough of them. And so what do we do in this lab? Well, the mating chamber is going to represent sex from generation to generation. And so what we're doing is putting all our alleles inside here. It makes what's called the gene pool. And then we're simply pulling them out, and each of those pair that we pull out represents a new organism. And that sets the next generation. And so what are we studying in this lab? We're studying 4 things. First thing is just trying to hold Hardy-Weinberg equilibrium the same. In other words, we're trying to make sure we have large sample, it's random mating, no mutations. All of those 5 things are exactly the same. And we should see that those 0.5 values stay the same. In the next round, we do selection. What do we do there? If we pull out a black and a white, then we're okay. If we pull out a black and a black, we're okay. But if we pull out a white and a white, then that dies. You could say it's a recessive disease, for example. We remove that, and then we calculate the next generation on that. And so I don't want to tell you what happens, so you'll have to figure it out. The next thing we model is heterozygote advantage. An example of that might be sickle cell anemia. Sickle cell anemia is an awful disease if you're homozygous recessive for it. But if you're heterozygous, you're actually protected against malaria. And so heterozygotes actually are protected. And so that's an advantage. And so we model that and see what happens to our frequencies. And then finally we do genetic drift. And so genetic drift is some kind of an event. We reduce the numbers from a large sample size to a smaller sample size. And then when we're done with that, we figure out what happens once we return that sample size to a large number. And so those are the 4 things that we'll study in that. And basically we're trying to model what happens in a population as far as genetics go. And that's about it. And so hopefully that's helpful.
Yes, I know.